All right, good afternoon, everyone. This is Anindito Sen, or Andy, as people call me. Before we start, what do you see on your screen? Do you see just the single slide with the title of the talk? Is that the correct thing, Ashlyn?
Yes, Andy, you should be good.
Okay. All right. So what we're going to do today is that we will walk over a bunch of slides, which will let us know the new Cryo Dual Beam, or FIB, that we call, is a focused ion beam SEM. It's a high-end SEM that we are going to have at MIC in the next few months. We will explore the scope of this instrument, the application capabilities, and you can correlate to those as how you can use this instrument to enhance your research work and create awesome data. Kindly note, this is sort of dummies for beginners. All of these information is also available online. Much of those are also available on YouTube as well. And I will share those links with you guys. If you have questions, put on the chat window, as Ashlyn said, or send me an email and I'll reply to them as soon as possible. Okay, so what we're going to do here is we will see the outline of the instrument, the sample preparation equipments that we are going to get from Leica. We will see the data collection and image processing softwares and results and visualization. Now, sample preparation will not be limited to biological science sample. It will be also covering material science sample, and especially samples which are more important for nanotechnology, like drug discovery and medicinal values of those nanoparticles that people use nowadays. We will discuss a little bit on the conditions as how you should prepare your samples, what are the restrictions you may have while preparing it, and what are the instruments that's going to help you to do this. Obviously, for the data collection, now this is something that you have to do hands-on with the instrument, but we will have a bunch of different softwares. Amira and Aviso, Stack and Viz is also available, ImageJ, BSoft, iMod, EMAN2 is also available. So ImageJ, BSoft, iMod, these are all open source programs. And to best of my knowledge, Amira and Aviso is also written in Python. ImageJ is so as well. Leaving out BSoft, I believe iMod is also the same. So if you need some special filters to be put in, for example, if you need to apply winner filter with a baseline corrector filter, you can write that program in the Python code and implement it to the image processing code that we have. That is also available with us. Kindly note that you need to be very thorough when you're writing that program, and it is going to be very important. Do we have any questions coming in here? Oh, no. Okay. Give me 1 second. Yeah, chat. All right. Okay. For the results and visualization, and this is something really I'm excited about. We have all sort of different softwares that we know of, COOT, UCSF Chimera for 3D visualization of these electron density maps that's coming from FIB, Cryo-FIB, or TEM reconstruction using single particle tomography. But this time we are going to have a software package from Avia, which will be really good for the fact that you will have a machine learning instrument, which means that if you want to do a segmentation, you don't have to do the whole segmentation by yourself. You will do a part of it. In fact, say 10% of it. And the program will pick it up and it will do the rest of the segmentation by itself and give you the results. So it's going to save you a lot of labor, and it will be much more precise than while doing it manually over the whole data set. Additionally, there will be what we call is a pair of goggles and a hand controller. You will put on the goggles, and with the hand controller, you can literally see the whole density map in 3D. And you will be able to control the density map using that hand controller. And we like to call it an artificial intelligence system. So Dan Gostovich of Thermo Fisher says that this is the first Helios Cryo Dual Beam with artificial intelligence in it. So if you just go by the terms, we are the first in that case. All right. So let's focus a little bit on the layout of the instrument. What we're going to have. This is the cartoon diagram. You will have the SEM column is seen in the center. You will have the gallium deposition needle in there, the fib, which will cleave off layers of the top layer surfaces of the sample. You can cleave as thin as 3 nanometers thick. So basically what you're doing is that you take a T4 bacteriophage, if you can, put it out there, and then you can cut off the head of the T4 bacteriophage in multiple parts. That kind of thinness is available in this system. But primarily, what we do with this kind of systems usually is studying the tissue sections, the subcellular organelles, or very tiny regions or domains of material science sample, like soil samples, magnetic materials. Although, let me tell you that this instrument is not capable of handling materials which is very rich in iron, cobalt, or nickel, because those are all magnetic materials. Rest of the materials we will be okay to study with. Now, as I have told you before, it is going to be a cryo dual beam. So obviously it is going to maintain a temperature of say minus 170 degrees centigrade to minus 180 degrees centigrade. That will be cooled by the nitrogen gas that will getting inserted in the chamber of the SEM from a liquid nitrogen dewar, which will be placed just outside the system. Please note that this is a real great technology, because in a TEM, you have a very small space to maintain that kind of temperature. If you are using a single tilt holder, the holder has a liquid nitrogen dewar at the end of it, and it cools the system. And that's a very tiny little space for the grid. But in this case, the whole system needs to be kept cold at say minus 170 degrees centigrade, at least the stage. And you have a very big chamber, the SEM chamber. So the technology has improved immensely over the last few years. And we were able to now have a temperature as close as minus 170 degrees centigrade or to minus 180. Now, while we are doing cryo work, kindly note that all your samples needs to be pre-processed in cryo conditions. So we will have a bunch of different instruments from Leica that will allow us to perform those functions. So you see the cartoon, there is a thing called cryo shuttle and a transfer rod. What happens is that transfer rod is attached to a liquid nitrogen dewar, which keeps it cold, and then we push it into the quick loader. And with the rod, you can twist it and place your sample onto the cryo stage in the SEM. Just to give you a bit of an idea in real space or real time, this is what the rod looks like. It's the quick autoloader. You will have these instruments for Leica. You see this hammer-like thing, which is actually the transfer shuttle, but all of these instruments will be used for cryo sample preparation. Now, those who do not want to do cryo sample, they can very much deal with regular temperature sample also using this instrument. So there is one question from Jin Hoi. So you mean the sample is already frozen before it is loaded? Yes, Jin Hoi, it is. So if you want to deal with a cryo sample, that is correct. You will pre-process the sample in liquid nitrogen temperature, or rather say minus 170 to minus 180 degrees centigrade. You will also have the option of going to say minus 70 degrees centigrade. If you are having a sample like a rubber, and I'm going to show you an example. Rubber doesn't like to be, say, at minus 170 degrees centigrade. It forms artifacts on there, but it is very happy to be at minus 70 degrees centigrade or minus 80 degrees centigrade. You have that option too. And I will show you a nice little data set that we did with the Hillsborough guys of Thermo Fisher up in Oregon in their factory. All right, so and the final part, that the artificial intelligence software that I was so much talking about, this is an example of such a segmentation. This, I think, is a neural network, and we will see a little bit more in the penultimate slide of my presentation. So many of the slides you will see will be from Thermo Fisher Scientific, from Leica, from EDAX, which I haven't talked about as of yet, and Avia, the software package. Okay, so let's talk a little bit about the workflow. Now, this is going to be very interesting. We will have multiple recipes of the workflow. What it means is that when you come with your sample, you will set your instrument in your file or in your account on the system computer, set up the different parameters you need to use for collecting the data. And we will save that. We call that a recipe. Okay, once that recipe is set, next time you come in, you don't have to play around with the parameters. You will just, give me 1 second. I think somebody is messaging me. Okay, so one of the things that, as I was telling, sorry, I lost myself. So once you have put the sample in the system the second time, you do not have to play around with the parameters. You will just use the previous setting, and the instrument will automatically take it up from there and will give you the result. So that is what we call a recipe. Now, in the first column, as you see, you can prepare the samples as a normal fixed sample in a resin. You can do some ultramicrotomy, or if you have a material science sample, for example, soil, rock, or something else, or some metalloids, you don't have to do anything of these kinds. You have to do the milling and the polishing that you usually do for regular SEM. You will load it into the small stubs. There are multiple different stubs in there. You can transfer it to the system directly if that is not requiring cryo temperature, or if you have a cryo temperature, you will use something like the Leica instrument that I have shown here, and then you will transfer it to the SEM. Now, the data acquisition. You see this video that I have collected from the YouTube. Now you see that this red beam is the etching layer, which is the FIB. It cleaves off the top surface, the SEM takes an image, and then the FIB beam comes in, again cleaves the top surface, and then the SEM takes an image. Now this is something, it goes into cycles. So you have a very thick sample that you cannot study in a TEM because it has a limitation of maximum thickness of 100 nanometers. You can go to a couple of millimeters here using this instrument. So you will be able to form the whole electron density map of that particular region, ranging to quite an extent. So this is what the snapshots will look like. And then what you are going to get is a stack of data. This is one of my data sets on biological science sample, but keep in mind that the stack of data that you are going to get is just not a few tens of images, a few hundreds of images. Those are in thousands of images. And a data set is going to run crazy, something about 2 terabytes, 3 terabytes, or 5 terabytes. If you really want to do a very in-depth analysis of a sample that is, say, a few microns thick. Usually the instrument runs overnight and it collects the data. So there is a question from Deborah, how long it takes to collect a stack of images? Well, Deborah, it depends upon how many images you want to collect. Typically for, say, 1,000 images, and if you are setting it to a thickness of, say, 10 nanometer slice, then it will take about 8 hours or so. If you are going for 3 nanometer thickness, that is going to go much higher. And I believe it is not a linear equation. It is going to be a much more of a time than just multiplying it by 3.3 times of that of the time that is utilized for collecting 1,000 images for, say, a 10 nanometer slice thickness. Yes, so one of the interesting things, like I mentioned, is that you can collect the data, you don't have to sit in front of the instrument. That's good. You set it up, you put the whole system into your workflow, you have the recipe already done, and you leave. All you have to do is make sure that the liquid nitrogen devar is all good if you're doing cryo, or the basic things are there, like the supply doesn't trip off, the electric supply doesn't trip off. Okay, so, and once you leave, the instrument has this great capability that it will shut itself off when it reaches that particular amount of data collection. Hopefully everything going smooth. Okay, so if you have set it up, set 2000 images at this thickness, it will close the column, beam column, and all the other parameters and the instruments and the detectors once it reaches that 2000 slices that you have asked it to do. That's the beauty of this instrument. Now, then you will use different programs again, and then we'll generate something like this, which is a 3D electron density map. This is one of my previous work. And then obviously using those density maps, you have to do the segmentation, as I have shown you on the cornermost column. This is actually, I believe the top one is a nucleus of a breast cancer cell, if I recall correctly. And the bottom one is obviously a mitochondria where we have infected it with nanoparticles. So those yellow blobs that you see there are actually the nanomedicines. Okay, now let's look in a little bit more depth as how we prepare the specimen. Now we can prepare the specimen in multiple different ways. I have just picked up two here. You can do the chemical processing, or you can take the cryo processing. Now for the cryo processing, you can use the regular TEM grade, put the sample in there, and then use our newly purchased Cryo GP2 to freeze the sample by slushing the sample into liquid ethane at -180°C, and then transfer it to the cryo shuttles from Leica for conductive coating. Say the coating can be of gold, platinum, or carbon. Now you can do freeze etching, freeze fracture also. Now keep in mind that all of these materials that you will do for cryo are either hydrated or beam sensitive. So that kind of sample is always better to be prepared in a cryo condition. One thing that I usually come across that, okay, why the GP2 and why not a Vitrobot? Unfortunately, the Vitrobot has a problem that it blots from both the sides. The difference between keeping this sample, and I will show you in the next slide, sample is a little bit more thicker than a normal Cryo-TEM grade. Okay, so that's the reason why the Cryo GP2 will come handy. So it's a two-in-one instrument for the Cryo-TEM as well as Cryo-FIT. All right, once you have the conductive coating done on your sample, now for material science people, you don't have to do these. You can just mill it, clean it as you prepare your SEM samples for, and then use this transfer stage, or you can directly put it inside the SEM and set up the temperature for you. If you want the temperature, stage temperature to be -170, it is pre-cooled. If you want it to be on the cryomat, which is at -70°C, you can do that, or at room temperature as well. And just to distinguish between the two approaches, you will say that this is for hard materials, and for the soft materials, the lengthier process needs to be taken care of. Any more questions from anyone? Not at this stage. All right. So for the cryo specimen preparation, say using the Leica Cryo GP2, what do? This is the inside of the GP2 we have. Typically what we do is we will load the sample as you see in the first image, and there is the blot paper. And then the blot paper will simply come in the close vicinity of the filter paper, the sample that is deposited on the grid, and then there will be a blot, a normal blot. As you can see at the bottom image, just right to the mentioning of visualize in TEM. We will not do that kind of blotting. Okay, we will just do a soft blot. As you can see in the second line, we say soft blot force is weak. So we are going to leave a significant amount of sample on it, and we will plunge it into liquid ethane. Once that is done, keeping the grid cold and immersed in liquid nitrogen, we will transfer it to a conductive, cryoconductive coating in an like a coater, which maintains that temperature. The choice of the coating will be yours. You can do with gold, platinum, or carbon. Once that is done, you can load it to the SEM, a pre-cooled SEM, and that's how it's going to be. Now, let me show you an example of this. This is a sample that is provided by Paul Straight's lab in BioBio. This shows a cluster of filamentous bacteria. You can see the top and the side view. And in the bottom portion of the slide, you can see the platinum deposition area. So the conductive coating has been done. And then by milling, trenches are built, all right? So those trenches are the regions where we will focus in and collect images, all right? And kindly note, we are not going to use a very strong accelerating voltage to image this, all right? So then the purpose of doing a cryo imaging or using a cryomat imaging will be defeated. The purpose will be defeated, okay? So typically we use a 1 kV accelerating voltage to image that. But even that is quite good enough for us to get all the GUI details that we need to have from this these kinds of experiment. Okay, so once you are into the trench, you focus in there and you see these clusters of bacteria. You find the region of your interest. In this case, those buds are our region of interest. And once we reach there, we switch to a higher gear of magnification, we start cleaving in, and now you can see the inside details of the sample. Okay, and then obviously you will use your programs. You can have your own programs to process the stack of images and the data set, and then generate a 3D density map. So let me show you some 3D electron density maps that I have generated from this particular sample, and this is what it looks like. It is not the whole cluster of the bacterial cells. It is just a portion that I have reconstructed and have cleaved it off. Now the scale bar is there. You can understand how big of a region that we are talking about and what is the distribution. As you can see, it is pretty well crowded. And what you can do is that you can select out regions of interest, okay? So the regions of interest can be selected out. You can extract out one single filament and then look inside those cells for the more subcellular details. From this particular data set, what we were able to do is to measure the thickness of the cell membrane or the cell wall, and figure out like what is the variation of those cell membrane. Is it uniform, or there is something going on when there is a budding taking place or the cell division is taking place. So all those information is available to us. However, the information that is not available at this stage is the inside detail of the cells. So for that, what we do is we switch to the next higher magnification. We select an area and we start collecting more data to the higher magnification area. Now, while you are setting up the recipe, what you usually do, you collect the data set first time to do an overview of what the sample looks like. And then once you have a good idea, you do this reconstruction, you know what is the magnification that you want to select. You put those parameters into the newer recipe and you collect the data set. And that will be all. So from that point onwards, unless and until you want to do something different, you will be able to use that particular recipe to collect as much as data as possible. So here are two slices. I'm interested in the subcellular region, so whatever is there inside the cell. Here you can see there are two regions. On the top one, I see some very faint membranous structure, and the bottom one I see some globular protein molecules. Okay, so when I did the reconstruction, this is what it looks like. All right, so I see some densities as globular densities in the bottom portion of that particular bacterial cell region, and the top one has some membranes in there. Now, That is good, but one will always ask the question, these are filamentous bacteria, and my sample extends to, say, about 300 microns. How am I going to do it? Well, from the stack of images, you can generate the reconstruction, and then you can extract out the whole filament from that electron density map using computational analysis. And we have the software, Amira and Aviso, with us, and so you can use that to trace out the whole path of the filament. And so let's show you an example of that. This is a single frame, the same image, where you see some globular densities inside the cell. And this is what I did, is that I computationally reconstructed the density map of the whole filament, and then I extracted it. Now you can see an uneven distribution of those globular protein molecules. Okay, we do not know what the function is this, but there are multiple other things that you can study using this. Keep in mind that this is just a simple example. We have not done any further studies on it. So this is the kind of details you can get. Note, those globular domains are not more than 20 nanometers, which is very high in order of in terms of resolution for an SEM. Okay, all right. So what about material science sample? And this is, I get the fire every time when I speak that, well, about the biological science sample, and everybody will say that, what about the material science sample? You have not shown some slides, but we have done some work here also. Before going on, let me see, somebody has put a question. Julian Feng says, have you stained your sample? If yes, before freezing your sample or after freezing it? So Julian, we do not stain our samples if you're doing cryo, okay? So if you want to do staining, you have to stain the sample while doing the cryo. What we call is that is a cryo-negative staining. Please note that once you put the stain into the solution and you let it to dry, structural deformation, if you are really interested in having those things removed, will happen. So the staining, what we call is negative staining, negative staining in cryo, where we put this stain solution. Typically, it is about, let's say, 12% saturated ammonium molybdenate solution mixed with the sample, and then we will do that. So Daniel says this cryo-negative staining would be similar to negative staining for the TM. No, Daniel, there is a bit of a difference. So what we will do is that in regular negative staining for TM, we let the sample to sit and dry. Okay. What we do here is that just before plunging the sample blotted into liquid ethane, we will put the staining solution, which is ammonium molybdenate, say in this case, mixed with the sample on the grid. And then we will do a soft blot that I've shown you in the last slides, last couple of slides, and then we will plunge it into the liquid ethane. Okay, and then we'll follow the whole procedure. However, you can stain your sample, but that will not be a cryo sample. Okay, you can stain your sample. There are other methods where, in a way where you can stain your sample for longer time, and then you dry it. You can air dry it and use it as a regular fixed sample, like chemical fixation or something. All right. Does that answer your question, Daniel and Juan? Okay. So let's move ahead. So let's look here into this material science sample. Now here is a small piece of rubber that was submitted to MIC, and Rick Wilton here at MIC kindly provided me this sample, and we wanted to see, like, okay, how does the material science sample really perform in these instruments, because especially this is made for cryo. And the reason I picked up rubber is because rubber is soft. All right, and so if you try to do some sectioning on the rubber, it will not stand that force. So it needs to be pre-cooled at a given temperature, but rubber doesn't like to be cooled at, say, -170 degrees centigrade. So the optimum temperature for that is around about 65 to minus 85 degrees centigrade. All right. So the method, I'm not going to be speaking in details. It is just jotted down in 4 simple points. You can take the rubber and do all this pre-processing steps, and then you can set your stage at minus 70 degrees centigrade or max at minus 120 degrees centigrade. That's what we call is the cold trap. Once that is done, we will perform the same kind of image processing and image acquisition and image processing as we have done for the material science bioscience sample. Now here are some slides showing that you can see the platinum deposition is done at the room temperature. Again, there is nothing to do with cryo at this stage. This is an interesting thing. The platinum deposition or the conductive coating has been done in room temperature. And then you see the initial milling has been done at minus 160 degrees centigrade. Okay, so this is part cryo. The stage was kept cooled at minus 160 degrees centigrade so that the rubber becomes hot. And then you start sectioning. You can still go down, go up actually, from minus 160 degrees centigrade to minus 70 degrees centigrade or minus 120 degrees centigrade and start collecting data. Now, once it starts collecting data, you see the bottom 2 images, there are tiny spots of white densities in there among the whole gray background. And those densities were the region of interest for us. The scale bar is 5 micrometers long. So you can imagine one tiny dot, how small it can be. All right, so here it is. A little bit of the trench that has been built and the region where the data has been collected, a few of the parameters that we have used. Please note, in the previous slide, I've shown you the data was collected at 1 keV. Here, you can use 30 keV as an accelerating voltage, because this is rubber and the cryomet is providing enough stiffness to the rubber to be imaged. Okay, the fit current is about 0.3 nanoamperes. A slice thickness at this level is 10 nanometers, so really, really thin. The SEM voltage is 2 keV. SEM current is 25 picoampere. The SEM mode, well, it varies from instrument to instrument. In Thermo Fisher, they call it the opti-tilt and a few other things. The cryo temperature was finally set to minus 70 degrees centigrade. All right. So on the right-hand side, at the bottom right, you see some yellow densities. Actually, those are the densities which is represented by the white patches in the second row. And then we collected a stack of images and I did a reconstruction again. And this is what it is. It is actually a movie. Let me start it. Kindly note that I have inverted the density of the whole image sets here. So this region of interest, I'm not sure if you can see my mouse here. These are the regions of interest. All right. So let's just run this. As you can see, we are going slice by slice. And the bottom, I show one single slice. Again, the density is inverted here. And with this data set, I did a reconstruction of the whole rubber that we have introduced into the SEM. And this is what it looks like. You can see the distribution of those tiny little densities. We are not sure what these are, but to me, it appears it may be sulfur deposits. But typically, we do find such sulfur deposits in the in rubber. So we'll want to look into a little bit more details. This is what a close-up view looks like. If you see those tiny little blobs, those range from a size of say 10 to 50 nanometers. So this is the smallness that you can get in. I'm not even sure if smallness is a correct word for this, but this is the resolution that you can get using this instrument. Believe me, we are using an SEM, and that is very, very good for an SEM resolution. Okay, any questions that anybody has at this time before I switch to the next portion of the presentation? Okay, all right, so on the top right, you can see a bunch of different samples that I have written. Hydrogels, rock, soil, mica, graphite, fibers from clothes and metals. These are already being studied extensively using this instrument or this kind of instrument. Now, one of the interesting things that this instrument can really work with is specimen preparation for TM tomography. Okay, you can prepare ultra thin specimen and use that in TM for tomographic analysis. So in a sense, you are avoiding doing sections in an ultra microtome. Okay, the whole thing is automated. In ultra microtome, you can get a thinness of something like 80 nanometers. But here, your thinness can be as thin as 10 nanometers. Technically, you can have 3 nanometer thickness. I'm not sure if somebody has ever done a 3 nanometer TM tomography of a sample. Okay, but what does it look like? Well, here is a cartoon diagram of the instrument accessory that we will have. There is an ultra thin needle which attaches to the sectioned portion of the sample, and then it is lifted up. For the Thermo Fisher instrument, they call it easy lift for cryo assembly. And what it really does, it is better to see it in a movie. And the next slide is going to do exactly like this. Now you see here, the central portion is there. That yellow circle is showing you that. And that is the portion which is attached to the ultra thin needle. And the user is actually lifting that section region from the sample. And on the right-hand side of the movie, you can see another view of it. And then he is slowly controlling the lift. Keep in mind, the stage temperature is minus 160 degrees centigrade. So everything that you're doing is at minus 160 degrees centigrade, right? And so this is how the lift has been done. You pick it up and you can put it on the grid. And obviously the grid is pre-cooled, and you can maintain the temperature of that grid and transfer it to a Cryo-TEM autoloader grid for a Krios or an Arctic or an Glacios, or you can put it in to a cryo holder with a Gatan cryo holder. Okay, now this is what it looks like. Let's show you some snapshots of this. The top three shows the snapshots of the movie that I've shown you before. The bottom one shows the region of attachment of the sample and how does it attaches itself. Well, there's all different kind of techniques to that, and I'm not going into those details. And then you can slowly put it the slice onto a grid, and the last image actually shows that the cryo deposition less attached to the grid. Okay, there are other methods by which you can do this, but with slight differences. But typically, this is the baseline for carrying out these kind of experiments. All right, now what we're going to have from EDAX, we will have a 3D EDS software package from EDAX, and that is called 3D IQ, Team 3D IQ. Now, we usually, what we do is that we will take a 2D, we usually do 2D analysis or EDAX, EDS analysis of the specimen. Now, when you have a bunch of different slices from the sample that you have mechanically polished, and you have done the island milling and all those things. I'm not very good at this because I don't do these things much, but from my previous experiments and experience, I can tell you that when you have a bunch of images that you have to do, and if it's a whole thing is automated, you can generate the 2D maps as shown here. Okay, those are the different elements. Those are listed on the side. And what if we overlap all of these together to generate a 3D map? Now, you can use X-rays to do the same work, and the X-ray obviously is going to give you much more better of a resolution and information. And this is what the X-ray data set will look like. But there's a lot of different dots, and it is very hard to interpret. Now, if we use the FIB, you can generate density maps, something similar to this. Okay, that's much more easily interpretable, and then you can carry out your analysis the way it is. You have the particle spectrum, the cumulative spectrum, and then you can do your analysis. Of course, you have the option of putting whatever coloration to the peaks you like. One interesting thing, and this is what I've been told by the EDAX people, now Amatec, is what if I am interested in a particular region of that electron density map? So do I have to do computational analysis to select out that region? Technically, in many of the cases, the answer is yes. But in this case, the software automatically provides you that option. So you see this, there is a cylinder being drawn, and the user is interested in analysis of the region that is encapsulated by the cylinder. Okay, and that bottom portion shows that analysis. So you don't have to worry about the rest of the region. You can select out a region of your own interest. The program gives you that option on the fly, which means that if you have already done a reconstruction and your instrument is still continuing with collecting the data sets, you can take that the previously processed data set and do this analysis on the fly. And if you feel that, well, this is not going good, you can stop it, redesign your experiment, or prepare your samples and continue with this. Or if you feel that the results are really good, you can continue doing your experiments and the data collection, and it automatically gives you the results as you sit there collecting the data. So this is something really good. Now this is an example for modified steel. There are multiple different examples of this. As you can see now, there is the whole map of the EDS spectra coming from different elements, and a few more analysis have been done, but it's not just limited to these. You can do the cadmium, tellurium multi-layer structure, steel with corrosion. I have these examples in my next slide for the cobalt alloy and tungsten alloy. Now, let me show you those. Again, these are all provided to me by Amatec people. As you can see, these colorful images, these are just not colorful images. These are really informative images. And believe me, some of those have made the papers in which they are published really, really good, and went to very high impact journals. Do we have questions at this stage from anyone? If yes, please do put it in the chat, Zoom group chat. I don't see any. All right, so we will move ahead. Okay, what next? Now, what I have told you is the AVS software package, virtual visualization, something really great. It is like you are in the fourth dimension and things like that. But basically, what it really does, as I mentioned in the beginning, it has the ability of machine learning. Okay, there is a question from Deborah before I go. What is the resolution of the EDS volumes? Depends upon the thickness of your sample, Deborah. You can actually, so it will not be the paper spec resolution. It depends upon like how much volume of the data set you are collecting. So for example, if you have a very thin sample and you have done the EDS analysis of few hundred slices, then your resolution, if not absolutely great, it will be better than when you have a whole big sample analyzed. That is because of the fact that more the sample or the more the sample you cover and more images that you do, your resolution tends to drop a little bit because of the misalignment and other stuff. But theoretically, it also depends upon how well the boundaries are of your different domains of the different elements that you are studying. Does that answer your question, Deborah? Okay, if not, all right, great. So, okay, so as I was saying, the AVS software package. Here is an example, a typical example, machine learning. Now, this is the electron density map that has come from the FIP. Now, you want to segment these blobs. You have done a few within that area, and then ask the program to learn and do the segmentation for you. Okay, you just wait. As you have done a few for the machine learns it, and then there you go. It segments the whole density map for you. You may have done like 10% of that whole density map, and then the machine learns the process, it will do it. You can also define the parameters here. You can define the grayscaleness. You don't even want to do it. Like you don't, if you say that we do not want to do the segmentation, we just want to give you the parameters based on which you will do the, so say flooding the image or background calibration and stuff. program will do it. Okay, there is a question from Bilal. Can we obtain information about the roughness, RMS surface with reconstructed 3D images? Well, to an extent, yes, Bilal. So when you say the roughness, you have to have a scale to compare it. Okay, so if you have one sample and you want to measure the roughness of that sample, the RMS or the surface area with respect to some other standard sample, you can do that, but you have to have that, need to have that information. Does that answer your question? Okay, all right, so coming back to this machine learning thing, you can see how well it is performing, and there are multiple examples to these. So for example, here we have something, I believe this is from a leaf stoma area. I'm not 100% sure about this, but there are multiple other examples. As you can see, there is a neuron segmentation on the middle plane on the right-hand side. You can use this program or the software package for any image that you acquire from different instruments. It need not to be an SEM image. It need not to be an TEM image. Okay, you can use optical images also for segmentation. 2D segmentation, if you generate a 3D volume from an optical instrument, you can do that. Okay, so that is independent of, provided you should maintain the file formats that this instrument can read, but that is pretty much a versatile one. And so most of the very commonly used file formats are read by the software package. So you can literally do that. Okay, so it need not to be an FIB reconstruction, or it need not to be a TM tomography reconstruction. You can use light microscope or fluorescent microscope images also. Okay, all right. So just to give you an idea how difficult a segmentation can be and how useful the software will be, here is a volume, okay, that has been segmented. Just imagine if you have to do this manually. That is beyond somebody's imagination to do it, and he has to sit for hours and hours. I do not know hours or months or year to do this, but once you have trained the software, it automatically does the whole thing for you. To me, it appears like we are traveling through the universe or some you know, solar system, or let's say a galaxy. But that's what it is. You can see how well the pixels are well defined, how well they are separated, how well the grayscale regions are well separated from each other, weaker grayscale separated from the stronger values as well. Okay, and with that, I will just say thank you very much. If any questions, you can switch on your mics and let me know. Okay, so I'm gonna escape here and stop the screen sharing. All right, so let's go back. Okay, questions, anyone?
So I assume that auto segmentation, that there's some interaction there. It's not that you just do 10% and then you take whatever the machine spits out. That,
you can control that. You can control that.
That if the machine isn't doing it correctly, you have a chance to go back and redo.
Absolutely.
And not pick up some feature you didn't want.
Yes, absolutely. So what you can do is that, well, as the machine is giving you the output and you find that, well, that is not your region of interest or you're not happy with that, you can stop it right over there and teach the machine a little bit by doing another example or another couple of rounds of example, and then ask the machine to do the rest. Or if it fails completely, then obviously we go back to the same status where we do the segmentation manually. Both the options are available.
There's a chat question, Andy.
Name of the tool is Thermo Fisher Helios-5-CX. Well, yes, you will know it very soon. At this moment, I'm a bit hesitant to give out the whole details. questions from people in material science? Okay, so if you come across any questions that will be good or related to your studies, just email me that. Okay.
Good. Thank you so much, Andy. That was very informative.
Thank you.
All right, so be looking for October lunch and learns and October open houses, and I will send this out, the recording out later.
All right, thank you very much, guys.